Well, I'm going to go straight into it. Well, first, thanks for showing up in the morning and in the rain. So, some of you might be generally aware that I'm interested in understanding the evolution of single-stranded RNA viruses. Specifically, I'm interested in understanding the factors that influence the rate of evolution of these viruses in the wild, in nature, and understanding the factors that ultimately pose limits on the rate of evolution, and whether or not we can use and exploit these limits as a means of control. Now, the first question that you should ask is, why should we care? Why does anyone care about RNA virus evolution? And to that, the short answer is, it's because they cause deadly and sexy diseases. So, RNA viruses are ubiquitous, they're everywhere, they're diseases everywhere, and they have a huge impact in terms of mortality and morbidity. But also, they're really recognizable. You'll recognize some of these names: Ebola, dengue, influenza, rabies. So studying RNA viruses could be good for your career. But in all seriousness, a lot of the questions we have, a lot of the concerns we have about RNA viral diseases has to do with their evolution. We worry about them evolving resistance to the drugs that we treat them with. We worry about them evolving the ability to invade a new host, thus exposing an entirely new population, naive population to viral disease. So it's important to get a handle on viral evolution. And that's a practical reason for it. There are also more intellectual reasons for understanding viral evolution. Some of you were here last week for Katya Kola's seminar, and she mentioned that RNA viruses evolve extremely rapidly, orders of magnitude higher than DNA-based organisms like bacteria or anum. And so they evolve so quickly that you can actually see molecular evolution at observable timescales, which is rare in a lot of evolution. They evolve quickly, so quickly that we sort of think of RNA viral evolution as happening on ecological timescales. So, RNA viruses are a case in which we can ask and address questions about the interaction of ecology and evolution. So that's very interesting. The reason why I personally find them fascinating is what I like to call the filter process. Evolution, mostly, consists of the interaction of two processes. There is a process that generates variation, and then there's a filter process that eliminates most of this variation. What little of it makes it through the filter, we count up and we call the measured rate of evolution, and we use that to infer something about the nature of the filter. In RNA viruses, the variation generating process, for the most part, is mutation. Mutation caused by the extremely error-prone RNA-dependent RNA polymerase enzyme that viruses use to replicate with. And the filter process, unlike a lot of evolution, is more than just selection or drift. Now, we know that most mutations are bad, they're deleterious, and yet RNA viruses seemingly tolerate these extremely high mutation rates. They get away with it. So it's important to understand how they do that. And it turns out that they do it through a really clever combination of replication and demographics. And RNA virus replication in particular is extremely fascinating. And that is essentially what I'm going to talk about for the rest of this presentation. So I'm going to broadly divide my talk into two parts. I'm going to first talk about virus extinction, so the upper limits of RNA virus evolution, and how we can exploit that as a means of control. I'm going to talk about that through the lens of this phenomenon that Les mentioned, called lethal mutagenesis. I'll tell you what that is in a minute. And then I'm going to talk about what's happening in nature. So the various factors that affect the rate of evolution of viruses in nature. And in particular, I'm going to talk about how genome architecture affects that, and then how replication and demographics affect the rate of evolution. So let's go straight to it. So what is lethal mutagenesis? Now, really simply, you can think of lethal mutagenesis as a sort of shift in the way we think about viral control. Traditionally, when you want to control a viral disease, we treat it with a drug or some drugs that prevent it from replicating. Now, lethal mutagenesis is the idea that instead of worrying about preventing the virus from replicating, you prevent it from replicating properly. So in essence, it's instead of attacking the replication process, we attack the fidelity process of how accurately it replicates. Now, I just mentioned to you that RNA viruses have these extremely high mutation rates, and they seem to be doing okay. So why would something like lethal mutagenesis work? Well, we've seen over the last few decades that artificially increasing the mutation rate using chemicals like ribavirin increases the mutation rate of lab populations of viruses. Not too much, just 3 or 4-fold. That can cause extinction in a few generations. Now, when I say generation, now and the rest of this talk, I'm referring to a cell infection cycle. So a virus enters a host cell, replicates within the host cell, and then releases daughter viruses outside the cell. So that's what I refer to as one generation. So increasing the mutation rate of lab populations of viruses, just a little bit, 3 or 4-fold, can cause extinction in a few generations. And just for context, a DNA-based organism like E. coli can tolerate a 100-fold increase in mutation rate and still be relatively okay at the end of, at the end of the treatment. So clearly, viruses are somewhere different in terms of how much mutational tolerance they have. Right. So, what exactly is happening here? Even though we've demonstrated lethal mutagenesis over the last few years, the exact mechanical and theoretical underpinnings behind the phenomenon are still poorly understood. So, a few years ago, Jim Bull came up with this really elegant and beautiful explanation for what was going on. So Jim framed lethal mutagenesis as a demographic extinction process. And he framed it around the concept of the zero class. So basically what that means is, if we assume that the number of mutations per generation is Poisson distributed, this is an assumption we make all over the place. If you assume that the mutations are Poisson distributed, we can also assume that there's some fraction of individuals produced per generation that don't have any lethal mutations. And that fraction is described by this term over here. And you multiply that fraction with the total fecundity of the virus, the number of viruses produced per infected cell, NC, and you get the total number of viruses produced per cell that don't have any lethal mutations. So this is the zero class. So what Jim suggested was, if the mutation rate of the population is high enough that it brings the mean size of the zero class below 1, that's when extinction happens. And it makes sense. It's a really intuitive concept. Supposing you have an infected cell, but the viral population inside the cell has such a high mutation rate that all the viruses it produces are rubbish, then it's not going to infect any more cells. And extinction chains are going to get extinguished, and the population is going to go extinct. So it's a really simple and easy to understand concept. And from this mathematical formulation, we can understand that as long as the lethal mutation rate is greater than the natural log of the fecundity, that's what you need to do to prevent replacement. Or that's what you need to do to push the population to extinction. Right. But, there's a problem. It doesn't work. It doesn't work. The formulation has very, very poor predictive power. And Jim himself admits it in this paper he published this year in the fall in AmNat. And so if the point of this whole process is to make, is to model lethal mutagenesis in an accurate way, to use it ultimately for treatment in living hosts, then we need to be accurate and we need to be able to have some predictive power with our models. So what exactly is going on? Why does Jim's model sort of fall short? So the way I look at it, it has two major failings. First is there's a lot of ambiguity in the concept of the zero class. And the second one is that it lacks biological realism. He's made a lot of simplifying assumptions that don't quite hold up in the case of viruses. So first, let's unpack what's going on. Let's unpack the term for zero class. So this is a product of a fraction and a number. And so to bring this entire term below 1, you can either have an extremely high mutation rate and a high value of fecundity, or a low mutation rate and a low value of fecundity. And what I'd like to assert now, and what I'll hopefully prove to you over the next few slides, is that these two situations are not the same. There's a long-standing result in branching process theory that says that if you can assert, like here, that for a particular population, replacement on average is prevented, then you can also assert that that population will go extinct with probability 1. However, that gives you absolutely no information on how quickly that's going to happen. So that population could go extinct in 1 generation or in infinity generations. So that's not very useful. The other problem we have over here is in this term UC. So UC is the number of mutations per genome per generation, and that itself is a product of two terms. It's the product of the biological mutation rate, the mutations per genome per replication, and the number of rounds of replication within a cell, of which there could be many. And now we multiply this by twice the number of rounds of replication, because each round of replication involves first the production of an opposite sense template, and then the production of daughter progeny from that template. Remember, we're talking about single-stranded RNA viruses, so they need templates to replicate. Now, I think ignoring templates is a big mistake, because templates act as mutation accumulation bottlenecks. Now imagine a cell, a virus infects a cell and produces an initial opposite sense template. If that template has a lethal mutation, what happens downstream is irrelevant, because they're all going to inherit the lethal mutation. So not accounting for that template is a mistake. And I'll show you how big a mistake it is in the next few slides. So we figured out what was broadly wrong with Jim's model. So what I decided to do was explicitly model the within cell replication process. And I modeled it as a branching process, specifically a Walton-Gaston branching process. I'm not going to go into the details, because it's not important. What I'm going to tell you about is the sort of parameters we looked at. So we assume that there's some target fecundity within the cell, that's NC, and that fecundity is reached using some initial number of opposite sense templates, that's X, in some T rounds of replication. And in each round of replication, a single virus acts as the ancestor for G daughter progeny. So that's the general setup. We know that the number of rounds of replication in the virus, the mode of replication varies. It actually there's considerable variation in the mode of replication in viruses. It can go all the way from linear, where you essentially have a single virus enters a cell, produces a template, and all the daughter progeny that are released from the cell are copied directly from that initial template. That's linear replication. On the very other end, we have binary replication, in which replication happens over multiple rounds. And in each round of replication, the population size doubles and doubles until some target size is reached, and then the viruses are released from the cell. We also assume that there's some probability distribution associated with any one viral particle acting as an ancestor for the next generation. It's not, they're not always going to be copied. So we did a bunch of math, and we came up with this term over here. And this is the probability of extinction in one generation. Again, don't worry about this. I'll tell you what each of the parts mean. So the probability of extinction in one generation is equal to, roughly, the probability of a viable template times the probability of extinction given a viable template, plus the probability of a lethal template times the probability of extinction given a lethal template. And if we can assume that even a single viable viral particle produced per cell is sufficient to perpetuate an infection chain, then we can treat this term as a rate term. So it's the rate of extinction. And so this assumption that a single viable particle is sufficient to perpetuate infection chains is common, and that's what Jim uses as well. So now we have a term that tells us how quickly things are going to go extinct, as opposed to whether or not something is going to go extinct. So the first thing I wanted to look at was, does it matter? Is there a difference in the rate of extinction under various conditions that satisfy Jim Bull's extinction condition? Because if there isn't a difference, then perhaps his way is best. So what I've done over here is I've plotted the rate of extinction versus fecundity, and the rate of extinction versus the number of initial templates. And at all points in this graph, all points in the black line, Jim's condition is satisfied. So replacement on average is prevented all along this line, So what you see over here is that as you increase the fecundity, preventing replacement is associated with a higher rate of extinction. So preventing replacement for a high fecundity virus leads to like a really, really high rate of extinction. These things are going to go extinct really quickly. As opposed to preventing extinction for a low fecundity virus, it's associated with a lower rate of extinction. That said, this is not a huge range. This is 85% to something like 100%. On the other hand, when you look at templates, the story is a lot starker. So as you increase the number of initial opposite sense templates required for replication, the rate of extinction drops rapidly. So even going from one template to two templates, you see a massive drop in the rate of extinction. So remember, at all points along this curve, Jim's extinction condition is satisfied. Jim does not make there's no way to account for templates in his model. So when you actually do account for templates, you see that there could be a huge effect. In fact, when you have about 10 templates for a fecundity of 1,000, which is, by the way, not a really out there number, this is kind of realistic, you have a very, very low rate of extinction. So what you could have over here is a virus under an extremely high mutation rate that does really fine for a long time. So, and I think, my personal opinion is that this omission, Jim's not looking at templates, is really why his model has such poor predictive power. Right. So now I told you a little bit about how there's a difference between the rate of extinction versus whether or not something will go extinct. So I wanted to look at what happens to the rate of extinction under conditions of linear replication. If you remember, everything happens in one round. So over here, I've plotted the rate of extinction versus mutation rate, and I'm looking at different values of viral fecundity. And you see, sure enough, as you increase the mutation rate, things go extinct quicker. That makes sense. And there's some effect of fecundity, so there's a little bit of difference between having a low fecundity virus and a high fecundity virus, but there's not that much difference. On the other hand, if you look over here, these different lines indicate different numbers of templates, same thing. The rate of extinction versus the mutation rate per generation, you see a rather large effect of the number of templates. At low mutation rates, there's not much of an effect, but as you get into a higher mutation rate, so these mutation rates are where we think extinction will happen, there's a huge effect of the number of templates. And and it looks like, especially at a really high number of templates, that having multiple templates mitigates the effect of a high mutation rate, and maybe it allows viruses to be more tolerant to a high mutation rate. So I think it's important to get a handle on what's happening over here, like how many templates a virus uses to replicate. That's just that. Then I wanted to look at what happens under nonlinear replication. So when you have more than one round of replication, so I looked at the rate of extinction versus the mode of replication going all the way from linear to binary. So binary is the most rounds of replication. I did two things. I looked at it in two ways. Over here, I kept the biological mutation rate constant. So as you increase the number of rounds of replication, the effective mutation rate per generation increases. And over here, I kept the mutation rate per generation constant. So as you increase the number of rounds of replication, the biological mutation rate proportionately decreases. Over here, you increase the number of rounds of replication, the overall effective mutation rate per generation increases, and sure enough, the rate of extinction goes up. And there's some effect of fecundity, but to be honest, the pattern remains the same. There really isn't a huge effect. What's interesting is over here. So over here, I've kept the mutation rate per cell constant. So if you would measure the mutation rate of this viral population, it would still be constant. But you see some variation in the probability of extinction, and that variation is entirely due to the mode of replication. But here's where it gets really interesting. As you increase the number of rounds of replication, the probability of extinction goes down first, hits some bottom, and then it starts topping up. It starts going up again. So what's happening over here? So as you increase the number of rounds of replication, as I said, I proportionally reduced the biological mutation rate. So that makes sense. It's going down as we increase the number of rounds of replication. However, at some point, the effect of reduced biological mutation rate is superseded by increased template events, increased bottlenecks. So each round of replication involves another bottleneck. So if you have 2 rounds of replication, that's 2 bottlenecks. So at some point, the effect of bottlenecks supersedes the reduced mutation rate. So what that means is for any given mutation rate, there is some non-linear mode of replication that's best for the virus, because that minimizes the risk of extinction. And that could explain why viruses have mutation rates that are not purely linear or purely binary. They have something in the middle. This could be a reason why. Right. So that's all I have for that little section. I want to give you a quick recap of what I talked about. At mutation rates that are high enough to prevent replacement, on average, the probability of extinction is 1. However, that does not tell you how quickly things are going to go extinct. Given some mutation rate per generation, there is some non-linear mutation rate, there's some non-linear mode of replication that minimizes the risk of extinction. This is the best, I guess, mode of replication for the virus, or the safest. And then multiple templates greatly mitigate the risk and rate of extinction. And this, I think, is probably why Jim Bull's formulation has somewhat poor predictive power. And then finally, his formulation suggests that fecundity has a big role to play. I would argue that it's in fact replication and templates that have a big role to play in whether or not something will go extinct. All right, so I want to move on to the next bit, and that is looking at what's happening in nature. So the rate of adaptive evolution of viruses in nature. And before I go any further, I want to introduce you to a mathematical framework for looking at the rate of adaptation in asexuals. Again, don't worry about the math, I'll explain what the bits mean. So here's the basic idea behind this. There's an assumption that adaptive evolution, or the evolution or substitution of beneficial or adaptive mutations, is largely confined to genomes that are free of deleterious mutations. So this is in the case of non-recombining asexuals. The reason why we think this is true is because, on average, deleterious mutations are both more common and of larger effect than adaptive mutations. So if an adaptive mutation and a deleterious mutation co-occur in a genome, most likely the deleterious mutation is going to swamp out the adaptive mutation and it's going to get lost from the population. So yeah, again, we're assuming that there's very, very low levels of recombination. And this framework comes from Alan Orr's work from 2000, and so this is what it looks like in math. So here's what it means. There's two parts to that formulation, and that says that the rate of adaptive evolution Ka is proportional to the number of new adaptive mutations arising in the population per generation on genomes free of deleterious mutations times the probability of spread and fixation. And then, because this equation is set up in this way, you can actually look at the variation in the rate of adaptive evolution with the rate of mutation. And what we see is, as you increase the mutation rate, the rate of adaptive evolution increases up until it reaches some maximum, after which any increase in mutation rate beyond that optimum causes a rapid drop in adaptive evolution. And the reason why this is happening is because at mutation rates higher than the optimum, there simply aren't enough genomes available on which adaptive evolution can occur because they've all got deleterious mutations. So the way we can think about this is the rate of adaptive evolution is limited by the deleterious mutation rate of that population in the context of its environment. So what does that have to do with genome architecture? Well, it all comes down to sort of simplifying assumption that Orr makes. And what he does is he sidesteps the issue of whether he's talking about evolution at a particular locus or evolution over the entire genome. In the real world, we measure evolution by gene. And the rate of evolution of one gene could differ from the rate of evolution of another gene on the same genome. So the question is the rate of adaptive evolution of a particular gene limited by deleterious mutations just on that gene, or by deleterious mutations on other parts of the genome? So in other words, does location matter? Or specifically, does the location of highly conserved genes matter? So we know that most viruses have highly conserved genes. These are genes that code for very important functions and functional enzymes. And most likely, mutations in these really conserved genes would mess up the function of the gene. And therefore, these genes can be thought of as having an extremely high deleterious mutation rate. So the question is, does the location of these genes matter? Well, luckily for us, viruses come in segmented and unsegmented varieties. So segmented means a virus that has, where the genome is in multiple little bits, it's coded separately and then packaged together and sent out into the environment. And then unsegmented genes are where the entire genome is one long strand of RNA. So this allows us to set up a basic hypothesis to test. Now, what this is really about is strength of linkage. We're worried about how strongly linked the deleterious mutation and an adaptive mutation is. So now given that we have these two systems in viruses, we can ask this question. Now, if we assume that location does not matter, then the null hypothesis is that the rate of adaptive evolution of any gene is limited by deleterious mutations on other genes, both in the case of segmented and unsegmented viruses. Because if location doesn't matter, then it doesn't matter if the deleterious mutations are strongly or weakly linked. On the other hand, the alternative hypothesis is the rate of adaptive evolution of any gene is limited by deleterious mutations on other genes only in the case of unsegmented viruses, because they all occur in one single strand. So we got a bunch of data to look at this, and we got data from two viral systems. We had dengue, which is an unsegmented positive sense single-stranded RNA virus, and H3N2 seasonal influenza, which is a segmented virus. And from both viruses, we selected 4 genes each. From the literature, we selected 2 genes that we expected to be under relatively low constraint, or maybe perhaps even under selection for increased variation. And these are the genes in black. And then we selected 2 genes that from the literature we expect to be under a very high constraint because they code for important proteins or enzymes. And these are the ones in red. So in dengue, we selected the envelope protein and the NS1, which is a secondary antigen. And then the functional important conserved proteins were protease and polymerase. And in influenza, we selected 2 surface proteins as our less conserved proteins, and we selected a polymerase and a nuclear protein as a highly conserved gene. And there's plenty of support in the literature to suggest that these are in fact highly conserved and less conserved. Yeah?
Can you just clarify segmented versus unsegmented?
Sure. So, like I said, there's 2 kinds of genome, there's 2 types of genome architecture in viruses. You can have segmented viruses where the genome is coded separately in separate little bits. So you have small segments of RNA that are coded separately and then packaged together in one protein and sent out into the environment. So that's a segmented virus. So these segments can assort within a cell. And then you have unsegmented viruses where the entire genome is coded in one shot, in one long strand, and then packaged into a protein and sent out into the environment. So the major difference between the two is that segmented viruses, the segments can assort. They can get shuffled around. Oh, and also, now that you mentioned it, these 4 genes occur on 4 separate segments in influenza. Right. So the data we collected, we had a bunch of sequence data from GenBank, and these were gene and population-specific sequence data, and we got these were from human and host, from the Americas, from Asia and Australia, and we got 100 gene and population-specific estimates of substitutions per nucleotide per year. So slightly different from substitutions per genome per year, but that's not important. The difference is not important. And we used a phylogenetic technique with dated tips to estimate the rate of substitution per year. We also partitioned the rate of substitution into codon position, and I'll explain why we did that a few slides from now. Right, so let's look at the results. So over here, I've plotted the rate of substitution for these 4 different genes in dengue, and for the 4 different genes in influenza. And if you remember, the genes in red are what we expect to be conserved. Straight up, you can see that these two things are different from each other fundamentally. These genes don't appear to be very different. The rate of substitution doesn't appear to be very different. The rate of substitution appears to be different. But let's look at it statistically. I did a bunch of parametric and non-parametric tests. I'm going to report the results of the ANOVAs over here. There was no effective gene overall in the case of dengue, and there was a significant effective gene in the case of influenza. And then we did some pairwise tests to look at pairwise differences between substitution rate, and we used the 2 key tests for honestly significant differences. Again, we found no pairwise differences between any two pairs of genes in dengue. And in the case of influenza, we found significant differences in pairs of genes when we selected pairs across groups. So there's no statistical difference in these within group pairs, but there was a difference in across group pairs. And perhaps that's what's driving the overall effective gene in the case of influenza. So it does look like there's some difference in terms of how the substitution rate differs from gene to gene in the case of this segmented virus versus this unsegmented virus. Now, I mentioned that we split up substitution rate by codon position. So for those of you who don't know, the genetic code is read in triplets. And for the most part, changes on the third codon, the third nucleotide of the triplet, doesn't lead to changes in the associated amino acid, but changes in the first and second nucleotide leads to changes in the amino acid. This is not a hard rule, but this is true for the most part. So, assuming that expressed amino acid changes are adaptive if they appear in representative samples like the ones we have in GenBank, and assuming that silent substitutions are neutral or nearly neutral, then we can use the substitution rate on the first 2 codon positions as a proxy for adaptive substitution rate, and substitution on the third codon position as a proxy for neutral substitution rate. And there's some support and there's some precedent in the literature for using these as proxies. Again, these are proxies. These are not exact rates, but they're much easier to estimate than traditional measures of amino acid changes, which is the DNDS ratio. It's really hard to get at. And these you can just pull out from sequence data that you can get freely on public databases, and it's really easy to estimate. So here's what happened when we split up the data by codon position. Essentially the same pattern as before, but starker. So over here, this is substitution on the first codon position in dengue, and the same in influenza. And you can see this is really flat. There's not very much going on over here. Now here you can really see them starting to diverge. The substitution on the first codon position in these 2 conserved genes, that we thought are conserved, is much lower than the substitution on these less conserved genes. And when we did some tests, there's absolutely nothing going on here. There's no overall effective gene. There's no pairwise differences in genes in the case of dengue. Over here, there is a significant effective gene. And when you had across group pairs, you found a significant difference in the substitution rate. Now the same thing over here. This is the second codon position. Just as before, absolutely nothing going on in dengue. In influenza, this time, we found there's an overall effective gene, and there were significant pairwise differences for all pairs of genes. So all of these things are different from each other. And to contrast that, there's nothing going on in the third codon position. There's no overall effective gene in influenza or in dengue. There's no pairwise effective gene in influenza or dengue. So this has interesting implications. So we said that we're going to treat the substitution on the first and second codon position as a proxy for adaptive substitution. What that suggests is that the rate of adaptive substitution could vary by gene in a segmented virus, but not in the case of an unsegmented virus. And what this means is that for a segmented virus, you could have some gene segments that don't have any conserved regions on them, and these segments would be allowed to adapt to evolve at a much higher rate. Now, all viruses are going to have some conserved genes. They're going to need things like polymerase, right? So the question is, will the presence of that conserved gene limit the adaptive evolution of the entire virus? Well, it appears as if it does in the case of these unsegmented viruses, and it doesn't in the case of these segmented viruses. The way I like to think about this is, if you are in an environment that only imposes purifying selection, as in your genes are sort of doing their own thing and you don't want them to change, then you could probably do okay as an unsegmented virus. But if you're in an environment that imposes directional or disruptive selection in addition to the inevitable purifying selection on your functional genes, then perhaps it might be better to be a segmented virus. And that's interesting. That shows us these different ways of adaptation for segmented viruses. And just to contrast, absolutely nothing is going on with the neutral substitution rate. Right, so I want to quickly recap what I talked about right now. So the presence of highly conserved regions or genes limits the rate of adaptive substitution on the rest of the genome, or the rest of the genome segment, in the case of viruses. Segmented viruses segmented viruses can have gene segments that don't have any conserved regions, and these segments can adapt at a higher rate. Segmented viruses are possibly better suited to dealing with environments that impose different modes of selection. Now, again, there's a big caveat here. We only have 2 viruses. We only have one unsegmented virus and one segmented virus, right? So this could all be just specific to influenza and specific to dengue. So we need more data. Unfortunately, it doesn't really exist. There isn't very much annotated data that we can use for this sort of analysis. But we're getting there. GenBank, which is a big public database, people keep adding sequences to that database, and maybe one day we'll have enough to compile a big data set for another set of viruses. But so far, this is what we have. Right. Now I want to go on to the last bit over here, and that is rate of adaptive evolution in nature and what replication and demographics have to do with it. Now, I mentioned Orr's formulation before. Now, this is a really nice mathematical framework, and it works really well as a general way of describing how non-recombining asexuals adapt. However, it's not so great for viruses. There's a couple of reasons behind that. Firstly, even if we assume constant population size, which is a reasonable assumption for short periods of time, this population term n doesn't quite capture the tiered nature of viruses. Now, we know that viruses enter cells, replicate within cells, and then get released into the environment, sometimes in cohorts. We also know that we don't expect selection to act on these viral genomes when they're inside the cell. So selection only acts on them once they're released out into the environment, and they're actually getting released out into the environment in cohorts. Now, this has an effect, and I'll tell you how. The other problem with this general framework is that Orr assumes that the probability of fixation pi is roughly equal to twice SA. SA is the mean selective advantage of a beneficial mutation. This approximation is based on some simplifying assumptions, which I don't think hold in the case of viruses. So that's something to worry about. Now, both of these things, both of these points have to do with replication and demographics. So what I wanted to do was create a general mathematical framework to look at the effects of replication and demographics and see if they had a big effect on the rate of adaptive evolution. If they didn't, then perhaps this simple framework is good enough. So here's what I did. Assuming short-term equilibrium dynamics, the total size of the viral population can be thought of as a product of the number of viral particles inside an infected cell, NC, times the number of cells, infected cells. So the number of particles inside a cell times the number of infected cells. Now, we're interested in adaptive evolution, so we want to know how many adaptive mutations are there. So again, assuming a Poisson distribution, this term gives us the number of genomes produced per generation that have one or more adaptive mutations, right? When the rate of adaptive mutation is really small, we can approximate this term as NC times UC times XA. So that's the fecundity times the mutation rate per generation times the fraction of mutations that are adaptive or beneficial. But like I told you before, we only care about genomes that don't have any deleterious mutations, because if they have a deleterious mutation, it doesn't matter if they have an adaptive mutation as well. So we add this term over here, which gives us the fraction of individuals that don't have any lethal mutations or deleterious mutations. So this entire term gives us the number of new adaptive mutations rising in the population per generation on genomes that have no deleterious mutations. Right? So that's a better term to describe what's going on in viruses. Now, the other thing we had a problem with, was the probability of fixation term. So that term comes from Motsuo Kimura's really famous result on the probability of fixation of small effect adaptive mutations. And so it's this term, and that's approximated as twice SA. And this approximation is based on a few assumptions. We assume that n is huge, and the initial frequency of new mutations is usually 1 over the total population size. This is not always true for viruses. Now, I mentioned that viruses are released into the environment in cohorts. And so what happens is, assume a virus enters a cell, creates an opposite sense template, and that opposite sense template has an adaptive mutation, then the entire progeny of that viral infected cell is going to have that adaptive mutation. So that's a much larger initial frequency than just 1 over the population size, especially if the total number of infected cells isn't very large. So we tried to model that based on our understanding of the possible distribution of initial frequencies of new adaptive mutations, and we got this term over here. Again, I'm not going to go into the details, it's not important. I'm going to tell you, we plugged it all in together, it's very impressive. I'm not going to talk about this. Let's go to results. So this is the probability of fixation versus mode of replication. So this is the mean probability of fixation of new adaptive mutations. As you increase the number of rounds of replication, you see the mean probability of fixation goes up. And the reason why this is happening is because to have the minimum initial frequency, that 1 over the entire population size, that adaptive mutation needed to have occurred on the very last replication event, just before a daughter progeny was produced and packaged into a protein. So any adaptive mutation that happens before the last replication event is associated with a higher than minimum initial frequency. So the more replication events you have in the cell, the higher the overall mean probability of fixation is. And so each of these lines indicates what's happening at a different mutation rate. And what you see over here is as you increase the mutation rate, this entire curve gets flattened. And at really high mutation rates, at the mutation rate of 5 over here, mutation rate per generation of 5, there really isn't very much difference between what's happening at linear and what's happening at binary replication. And the reason behind this is at really high mutation rates, you don't produce very many viral particles without any lethal mutations. So the difference between having a single copy of an adaptive mutation and having all viable copies have a copy of that adaptive mutation isn't very large. So in a sense, the mean probability of fixation of an adaptive mutation is a function of the fitness landscape, the mode of replication, and the deleterious mutation rate. So this is another way in which adaptive evolution could be limited by the deleterious mutation rate at just a different level, a different scale. So that was probability of fixation. I wanted to plug it into the rate, into our equation for the rate of adaptive evolution. All these graphs, the black lines indicate our projections, and the red lines indicate the projections from a simpler model like ours. On these two graphs over here, I've plotted the rate of adaptive substitution versus rounds of replication. And over here, this is the rate of adaptive substitution versus mutation rate per generation. You'll notice I don't have any y-axes over here, and that's because based on demographics, this entire curve gets shifted up or down, but the basic pattern remains the same. And I'll talk a little bit about what's happening with demographics. So in both these cases, I've plotted the rate of adaptive substitution versus rounds of replication. Over here, I kept the biological mutation rate constant. So increasing the rounds of replication means effectively increasing the per generation mutation rate. And over here, I kept the per generation mutation rate constant. So increasing the rounds of replication means a lower biological mutation rate. So what you see straight up is as you increase the rounds of replication and the effective per generation mutation rate, the rate of adaptive evolution increases, hits a maximum, and then decreases after that. So sort of recreating the idea of an adaptive optimum, even in our complex model. And that thing is that same phenomenon is reflected over here when I'm just plotting it against the rate of adaptive evolution. Sure enough, you see it reaches a peak and then drops off after that. Now what's really interesting is this graph for me. So over here, this is a constant per generation mutation rate. So there's considerable difference in the rate of adaptive substitution, and this difference is associated with the same mutation rate. So you can have a much higher rate of adaptive substitution if you have binary replication than if you have linear replication, given the same mutation rate per generation. So in terms of what we can measure, this point is the same as this point, but it leads to different rates of adaptive substitution because it means that there's something else going on in terms of replication. So there are a few things that I want to point out just overall. We almost always underestimate the rate of adaptive evolution if we don't account for replication and demographics. You can see the red lines are always below the black lines, just basically. The extent to which we underestimate adaptive evolution is greatest at or around the optimum. Now, if we believe that viruses in the wild are near the optimum, and we actually have data, not mine, but it was published last year. We have data to suggest that viruses are near their optimum. Then that suggests that we're probably not getting a very good picture of what's happening, especially to the viruses in the wild. And here's where it gets really interesting. Under certain demographic conditions, especially when the number of infected cells is low, or at parity with the fecundity of the virus, and replication is nonlinear, the probability of fixation gets really high. It tends towards 1. So these conditions actually are similar to what we would see if you were looking at evolution at the scale of a single human host, especially early on in the infection. And I spoke to Katya last week, and so she talked about how some ways in which some broad vaccines that reduce the rate that reduce the mean fecundity of the virus could affect the rate of antigenic drift. And this essentially gives you another level of explanation for that phenomenon. Her phenomenon was what's happening between cells. This is what's happening within a cell. You can actually have really, really large changes in the rate of adaptive evolution at these very, very specific demographic conditions that, which essentially are similar to what happens within a single host early on in the infection. So that's really interesting. That suggests that we could have really, really fast rate of small effect adaptive evolution, within humans or within a single host. That's not something we've considered really. And also, if you keep everything else constant, the same mutation rate can lead to vastly different rates of adaptive substitution because nonlinear replication amplifies the rate of adaptive evolution. Now, if you remember from the first part of this talk, I said that there's some nonlinear mode of replication that minimizes the risk of extinction. Nonlinear replication also amplifies the rate of adaptive evolution. So it sort of adds to more evidence, adds more reasons to believe that viruses could have some nonlinear mode of replication. Right. And that's just a recap. Said everything over here. So, I hope I've communicated the importance of understanding what happens within the cell, especially in terms of understanding viral evolution. It's important. We've talked about we've sort of constructed a best practices of sorts for viruses. We know what mode of replication is best for the virus. We know what mutation rate is best for the virus. We know what demographic conditions are best for the virus. And so altogether, you can think of them as a sort of multi-dimensional fitness peak. But whether viruses are at this fitness peak remains to be seen. We just don't know. We don't have the data for it. We need more data. We need more sequence data, better spatially and temporally annotated sequence data. We need more estimates of parameters. We need estimates of the number of templates used in replication, the mode of replication. And the irony is, we know how to do this. Like there was a paper published in the mid-90s by John Gray that sort of showed how to estimate the mode of replication for a virus based on lab data. We just don't collect it because we don't think it's important. It is important, and we need to understand this stuff. And we need a lot more parameters to make what I've talked about useful. And so it's important to know where viruses lie in this fitness landscape because right next to the peak, this fitness landscape peak is a cliff. And if we know where the viruses are, we know how much we have to push to shove them over the edge. And so my hope is that this work leads to more work and eventually a more evolutionarily informed way of dealing with viral control. And that's what I have for now. I want to make some acknowledgements. I want to thank my advisor Les, who's been great. He's sometimes it can be intellectually intimidating. The depth and breadth of his knowledge is kind of staggering. Our conversations about viral ecology will take a right turn into behavior and then a left turn into Vasily Kandinsky and Pueblo pottery and what have you. It keeps me on my toes. I like it. I want to thank the rest of my committee. They've been really nice. I do want to thank Dave specifically because he's been great. I know a lot of you are familiar with Dave from your Pop Gen class and you get intimidated by the wall of math. Don't be. He's really nice. If you have a question that's really challenging and interesting, you should go talk to him. He'll help you. He gets like crazy gleam in his eye when it's a really tough, intractable question. It's great. I want to thank the past and present members of the real lab. Our lab motto is Li esami no finisco no mai, which means the examination never ends. And I want to thank them for this really perfect combination of support and challenge. It's been great. I want to thank the Rodo lab and the Gerardo lab for keeping the modeler tied down to the real world. Sometimes we make models for perfectly spherical clouds. It doesn't work. And I want to thank Aditi, my wife, for putting up with me. Yeah. And that's all I have, so let's go to the Q&A.